Thank you for that lovely introduction, and thank you for remembering our dear friend John Milner. So I've been asked to talk about dietary patterns and health outcomes. I have no conflicts of interest to disclose at this time. So I was asked to talk about the methods behind dietary patterns and then to summarize the available literature with regards to patterns and health outcomes. We will finish this talk with data needs and research gaps. So there are many experts in the field of dietary patterns, and I've done my best to present all of the work that is relevant to this field, but I'm limited to 25 minutes. So I know there are many of you in this room that work on dietary patterns, and I'm trying to honor the totality of the work that has gone into patterns research. So let's start with the definition of dietary patterns. It's the quantities, proportions, variety, or combination of different foods, drinks, and nutrients in the diet, and the frequency with which they are habitually consumed. So why should we care about dietary patterns? Well, patterns help capture the complexity of the overall diet and its constituent parts so that researchers can relate patterns to health outcomes. In doing so, we can essentially deal with the known collinearity of foods and nutrients. The dietary patterns approach has advanced nutrition research by capturing overall food consumption behaviors and its relationship to health. We also have positive interventions with dietary patterns approach, so thinking about DASH and Predimed. And finally, patterns are essential to inform policy, and our most recent iteration of the dietary guidelines has taken a decidedly important focus on understanding patterns. But there are always some limitations to any type of dietary patterns. Excuse me. So patterns often rely on self-reported dietary intakes, specifically from food frequency questionnaires and 24-hour recalls. And we know there are known limitations with these methods for reporting energy intake. However, there are at least 2 studies to indicate that energy underreporting doesn't have a substantial impact on dietary patterns. The first that I'm referencing is work that came from our lab at Penn State, where we generated patterns including and excluding energy underreporters, and we were able to preserve the same patterns regardless of energy underreporting. Another report using cluster analysis with food frequency had the same associations where they were able to preserve the healthy pattern, but they did see some deviations in a mixed or Western pattern. So I think this is something that we need to keep in the back of our minds when we're looking at this type of data. Some other limitations to the patterns approach is there's many subjective decisions that occur along the pathway, and we'll talk about some of those as we go through each of the methods individually. Patterns are named by researchers. So what is a Western pattern in one cohort isn't necessarily comprised of the same foods that would be a Western pattern in a separate cohort. So there is tremendous variability from study to study. The patterns may represent only small differences in servings from foods. And finally, I think it's important to understand and explore how we can leverage the successes that we've seen from randomized clinical trials and apply them in real-world settings, because at this time it's really hard to get people to eat 8 to 10 servings of fruits and vegetables. So Sue Krebs-Smith wrote this lovely article in Circulation last year that helps describe all of the different types of methods. Basically, they can be broken down to whether or not they are independent or dependent of a health outcome. And by that I mean the independent methods are essentially a 2-step process. We derive patterns in step 1, and then we relate them to health outcomes in step 2, whereas the dependent methods use the outcome of interest in deriving the patterns. So we'll unpack these in the next couple of slides. So indexes and scores. This is a way that we can compare dietary intakes to some sort of a priori specified criteria. And you've heard of the Healthy Eating Index or the DASH Diet Index. So this is a report card for how close a group is to meeting a specified set of criteria. This is great because it allows us to compare different cohorts and different groups using the same exact rubric. So it's less subjective than other methods, with the exception of how the scoring algorithm is generated, of course. So I wanted to put this paper up as an example of dietary indexes. So there are actually 4 different DASH diet indexes, and you can see how they were generated. So the 4 methods are listed here. And this comes from Paige Miller when she was part of the NCI group. Across all 4 patterns or 4 indexes in men, there was a protective association with dietary indexes in colorectal cancer. But in women, there was only a protective association for 3 out of the 4 indexes. So I show you this to make sure that when you're doing this type of work, you're looking at all of the different methods within the same cohort. If these researchers went out and just looked at the Dixon method without looking at the other methods, they may have erroneously concluded that there was no relationship between diet and colorectal cancer, for example. Next, I'll talk briefly on special or selective diets. So this is a group of patterns where people selectively eliminate things from their diet, either for medical reasons or for personal choice. And I think probably the most common researched dietary pattern is vegetarian diets. So I'd like to show you an example of a meta-analysis on vegetarianism and cardiovascular disease mortality. So compared to non-vegetarians, vegetarians have a lower risk of mortality from ischemic heart disease, but not from circulatory or cerebrovascular diseases. Now moving on to factor and cluster analysis. Basically, the easiest way to describe factor and cluster analysis is that factor analysis groups foods together and cluster analysis groups people together. So if we in this room were a cohort, everybody would get a score on how well they identify with a certain pattern if we used factor analysis. Whereas if we use cluster analysis, we would each be in separate and distinct groups. This pattern over here might be Western, this might be the wine and salad group. So it's really a different way of looking at patterns, and it really depends on the research question that you want to ask, which of these methods you would use. But essentially, these are both data reduction techniques to take a complex diet and reduce it to a few latent variables that we can then relate to health outcomes. Both of these methods rely on input variables. And so by that, what I mean is the foods and the beverages and how we classify and categorize them to run them through these models. So do we look at pizza as consumed as a food, or do we look at it as its constituent parts? The dough, the cheese, and the vegetable sauce. So that's an important consideration, is what are the inputs, and that will change the outputs, certainly. So a factor and principal components analysis works to create a linear combination of input variables to reduce to explain as much variation in these input variables as possible. And as I alluded to earlier, there's a number of subjective decisions along the process. How are you going to input the variables, the foods? That's something we've already talked about. The rotation of the method, is it Varimax, is it Promax? The number of factors to retain, and we'll look at that and the factor loadings in the next couple of slides. So when someone runs a factor analysis, this is what an output would look like. And this is a really nice paper by Gleason et al. That was in JAND last year that talks extensively about the methods behind this. So when looking at factor analysis, researchers often use eigenvalues, which is a fancy word to just say a mathematical term that represents the amount of variance that that specific factor explains. So in this table, we have 8 input variables. So there were 8 food groups as an example. So you will see an eigenvalue for each of those principal components. And a general rule of thumb is anywhere over 1, you want to consider that to be a potential factor number. We also have the percent of variance explained by each of the factors that were generated, and of course a cumulative percentage that can help researchers decide how many factors to retain. Another way that researchers decide how many factors to retain is by looking at a scree plot. And this is a different data set than on the previous slide, but you can see that there are the plot of the eigenvalues on the y-axis, and the dashed line represents 1. So you can see that anywhere from a 5 to a 4 to a 3 factor solution would be defensible looking at this scree plot. And others would argue that you're looking for that big drop in the scree plot. So another researcher might look at the same plot and say, wow, we only have 1 factor. So there is a high level of subjectivity to how the number of factors are determined. The next step in the process is looking at factor loadings, which can loosely be, I guess, compared to a correlation coefficient, so it ranges from -1 to +1. And I wanted to show you an example from the California Teachers Study about dietary patterns and breast cancer risk. So in about 4,000 women, these researchers found 5 different dietary patterns and related that to risk of different types of breast cancer. So this slide is probably very hard to see, but I just wanted to point out a few things. One, I have circled at the bottom, foods and beverages with factor loadings greater than 0.35 are shown. So each researcher decides what is a meaningful cutoff for where a factor loading is important. So in this particular example, it's 0.35. Another researcher might use 0.2, 0.4. So that's important to consider. These researchers found that a plant-based pattern was protective with breast cancer. So I really don't know if you can read this, but you're going to have to trust me here. The plant-based pattern is comprised of peaches, strawberries, oranges, broccoli, bananas, what you would think of plant-based diet, right? So this is what they named the diet. But in this cohort, there was another pattern that was ethnic, that was also described of what I would consider plant-based foods, lentils, beans, tofu, rice. So you can see how the subjectivity and the naming of the patterns can lend itself to having some confusion in the field. We say a plant-based pattern is associated with reduced risk of breast cancer, but what types of plants? And that's an important point to consider, I think. Next, we'll move on to one of my favorites, cluster analysis. I like cluster analysis because it's really clean. You're either in or you're out, okay? And the way that the computer generates this is to keep the groups as similar as they can and be distinct from the other groups by as much as possible. So cluster analysis is really complicated, and I'm going to try to explain it. I said in a tweet, but there might be more than 140 characters here. First, you need to decide what is the measure of similarity. So what is the yardstick for how we're going to compare people to one another? You select the algorithm you want to use to design the clusters. You have to choose the number of clusters that you want, and then you have to interpret those clusters and name them, much like we did with factor analysis. And then it's always recommended with cluster analysis, because it's not as exploratory as factor analysis, that you validate your clusters somehow. So you can validate clusters by running cluster analysis on subgroups, by using simulated data, or by interpreting them. If you have a cluster that is high in dairy food, does that cluster have higher calcium intakes than other groups? So that's one way to sort of essentially validate cluster analysis. This is the distance measure that's typically used for the most part in nutrition research. It's called Euclidean distance. There are 2 main types of cluster analysis. But by and large, in the nutrition field, we've utilized the second method, the partitioning or the k-means, which basically assumes that we have some sort of prior knowledge to the data structure, which may or may not be true. So you have to tell the program where to start, and you also have to specify how many clusters you want the program to give you. So this is a study from a bright young graduate student in 2006. I was in EB, I was just young and so excited to be here. We were looking at that time at the differences between the input variables. So at that time, way back in 2006, people were really looking at the percent energy contribution and how that formed clusters. But at Penn State, the brilliant people that they were, they thought we should look at the number of servings, because fruits and vegetables, you can have a lot of fruits and vegetables, but really contribute very little to energy. So we did a comparative analysis looking at number of servings and energy contribution, and indeed what we saw with both methods is we had 2 clusters, each with a higher HEI score, but really only the number of servings was interpretable. So we had everything that you would think of, fruits and vegetables, lean meats. Whereas when we looked at the percent energy contribution, the group with the higher HEI score had more sweets and snacks and things like that that you don't associate with having a higher HEI score. So it's important to do different types of replications within your own cohort if you're going to be doing these types of methods. And that's exactly what Jill Reedy and the group at NCI did. So I just wanted to put this up there as a gold sticker for doing a really good job with the NIH AARP cohort. They looked at all of the methods that I've described to you and somehow got this all into one paper. It's amazing. So they looked at cluster analysis, factor analysis, and 4 different indexes. And what they found that in men, really, things were well preserved. Men tended to behave a little bit better or something. Fruits and vegetable cluster, there was also a fruits and vegetable factor. That and the reduced fat and diet foods factor were all associated with reduced risk of colorectal cancer, and the meat and potatoes factor was associated with increased risk. Whereas in women, they really only found an association for the healthy eating index score for protective association, and again with meat and potatoes for an increased risk. So the conclusion that these authors said is that while there were similarities found across the methods, suggesting that there were basic qualities of healthy diets that were preserved with the methods, there were still some differences, and that's because each of these methods answers a different question. They're all looking at patterns, but in a slightly different way. And so that is something that you really need to take home with you when you're interpreting dietary patterns and health outcomes. So I'm going to go through these next 2 very quickly because we haven't done a lot of work in the nutrition field with these 2. The first is reduced rank regression. It's very similar to cluster or factor analysis in that it employs those eigenvalues and eigenvectors to determine the factors. But in reduced rank regression, we were looking at a surrogate endpoint, a surrogate marker for an endpoint. And using that, we want to explain as much variation in that. So usually a biomarker or something like that. With classification and regression tree analysis, this is something that sounds so good on paper. So we have a disease, we have heart disease, and we want to look at the diet and explain as much variation in heart disease with the diet. Unfortunately, these methods were not developed for nutrition questions. That hasn't stopped us from trying. But I think there's some real potential here, but right now it's not really ready for prime time. So now what I'd like to do in the last 7 minutes and 46 seconds is summarize. we were like told we were going to get a beat down if we went over time, so I'm trying really to talk fast but to be effective. So thanks for the smiles, it's helping. So I'm going to rely heavily on the Nutrition Evidence Library from the USDA, the systematic reviews with 3 health outcomes. So the USDA, they looked at papers that had all of these indexes and scores, as well as cluster analysis, factor analysis, and reduced ranked regression. So the Dietary Guidelines Advisory Committee asked many important questions, but the first of which is, what is the relationship between dietary patterns and risk of cardiovascular disease? This was the body of evidence for which the NEL had provided. They provided a Category 1 strong evidence from 55 studies, 45 of which measured diet with food frequency questionnaires, and the majority of which were prospective cohort studies. And you can see that I've outlined the endpoints of interest and the number of studies aligning with those here. They found limited evidence for cluster and factor analysis, and this is because a lot of the reasons that I explained earlier, a lot of the subjective decisions, there's not a lot of comparability between studies. It was very hard to synthesize that literature, but they did have 22 prospective cohort studies. Here's what I mean when I talk about a limited body of evidence for reduced rank regression. There were only 4 studies, so there was no category that was assignable for reduced rank regression and cardiovascular disease. So the Dietary Guidelines Committee gave this a grade of strong evidence. They relied heavily on the Nutrition Evidence Library, and they also looked at the systematic evidence review from NHLBI, as well as the AHA and ACC guidelines. So their conclusion statement was, strong evidence exists for CVD for consuming a diet rich in fruits and vegetables, low-fat dairy, fish, whole grains, fiber, potassium, and other minerals at recommended levels, and a diet that is low in red and processed meats, sugar-sweetened foods and drinks, saturated fat, cholesterol, and sodium. The second question was about body weight and obesity. There was an NEL2 moderate level of evidence here, and that came from 14 studies. Again, similar issues with factor and cluster analysis, so limited evidence was able to be distilled from this body of literature, and again, only 6 studies with reduced rank regression, so not assignable. So the Dietary Guidelines Committee relied on this data as well as the other 2 references listed here and had a strong evidence for the role of dietary patterns in obesity and weight status. So dietary patterns that achieve energy deficit and operate as part of a comprehensive lifestyle interventions can result in clinically meaningful weight loss. And those patterns are high in fruits and vegetables and whole grains, moderate in dairy and alcohol, and low in meat, refined grain, sugar-sweetened beverages, and sugar-sweetened foods. Finally, what is the relationship between dietary pattern and risk of type 2 diabetes? Using scores and index, the NEL found there was limited information. Similarly for factor and cluster analysis, and not assignable for reduced rank regression. However, the Dietary Guidelines Committee elevated this to a moderate level of evidence based on a meta-analysis of 15 studies that indicated a 21% reduction in diabetes with a pattern comprised of whole grains, vegetables, and fruit, and a substantial increase in risk in type 2 diabetes for diets that had higher consumption of red and processed meats, high-fat dairy, refined grains, and sweets. So in concluding with these last few slides, I wanted to just talk about data needs and considerations. I think we're talking today about big data because these patterns are likely to track with other health-related behaviors. There's also emerging science looking at patterns in the microbiome. We need to, as a field, understand how patterns evolve across a lifetime, how childhood exposures increase the risk for later onset chronic disease in adulthood. We need to better integrate information on the timing, location, and social aspects of food consumption and dietary patterns. More research is needed, as I alluded to, on reduced rank regression and CART methods. We need to understand how we can encourage behavior change to leverage the success that we've seen from effective clinical trials with dietary patterns. We need to better understand how race, culture, and socioeconomic status influence dietary patterns. And finally, we need to keep hammering in those comparisons and validations that I showed you in a few slides. And there's an ongoing program called the Dietary Patterns Methods Project that is really doing this in a systematic way with multiple cohorts. And this was published last year in the Journal of Nutrition. So my key points: dietary patterns reflect habitual exposure to foods and nutrients, and I think therefore serve as an optimal method to assess diet and disease relationships. There's evidence to support the utility of dietary patterns for evaluating chronic diseases at this time, strong evidence for cardiovascular disease and weight status, and moderate evidence for type 2 diabetes. I think this platform today will leverage some new and exciting opportunities that we have in this field, and I'd like to thank you for your time and attention, and I have 2 minutes left, so please give me a question. Hi, Richard.
How are you doing? That was a great talk. A question about the clusters and understanding also aggregated, disaggregated food components, that sort of thing. How do you distinguish, how do you work with, say, the made-up person who just gets their fruit from jams, pies, whatever, and the person who gets their fruit raw, unprocessed, whatever you might, and equal amount?
Yes, so you can see there's a way of doing that when you take the foods and you look at the equivalence database. Mm-hmm. So the fruit and jam is like this much, and so it can either be represented as part of fruit, or it is much more likely falls into an added sugar category more so than the fruit that's in there, unless it's 100% fruit product. But you're getting like 1/17 of a serving or something like that. So it has a way of working itself out, but that is an important concept.
Hi, I'm Charlotte Wright, visiting from Glasgow in the UK.
Welcome.
I'm a great skeptic about this sort of big data approach, and I want to just raise that thought in people's minds that what you're describing really, although very robust mathematically, is essentially observational and qualitative, as you yourself acknowledge, the way the factors are chosen and described. And I think the worry is that because we put lots of dietary data in, we assume that what we're getting is something that is all about diet.
Well, that's what we're talking about here, is we're trying to move beyond just diet. And so I'm laying the framework of explaining how we have evolved from looking at traditional one nutrient, one effect, to looking at the totality of exposure. So I'm setting the stage for the experts here who will take that and explain how that fits into.
I suppose the point I'm trying to make is that the underlying truth that that factor might be describing is the cultural characteristics, the smoking habits, the many other habits that aren't in your model because you have.
I think we're saying the same things, is what we want to now expand diet and look at lifestyle, look at social behavioral things, look at everything, the patterns, if you will, instead of just focusing on diet. So I'm setting the stage in saying what has been done with diet, and we're going to build on that and talk about how we can incorporate that in a bigger context.
Brilliant.
Thanks.
Any other questions for Regan? If not, how about we thank Regan again one more time?